So I'm currently in Berlin in Humboldt University. So it's just very recently started my new postdoc there. But this work today I will present to you is actually done in the Free University of Berlin together with Audrey and Uli. I would like to acknowledge about that. So and we will go back a bit to bacteria. So and I slightly changed my title. I added the asymmetry in my title. I apologize from organizers, but I would like to thank organizers for this great event. And also giving me the opportunity to talk here. All right. So, and this talk is going to be mostly related to how and whether actually we can scale from single cell data, single cell behavior to population actually dynamics. So, and this is especially will consider sort of like a stressful environment when there is an aging, actually, there is a damage dynamics is playing a crucial role, right? So, and I will consider sort of like a single trait, the dynamics, also the damage dynamics, and try to understand the survival and the reproduction, and from there, try to actually scale to population dynamics. And then we will try to understand whether actually anything related to this damage dynamics, that is the stochasticity, asymmetry, etc., how do they actually shape the population dynamics? And this is going to be a bit a mix with the theory and the experimental study. So I will first try to go a little bit to theory side, and then we'll talk about the experiments. I don't see the time yet. It's maybe here. Perfect. But let me just first of all talk about sort of the theoretical framework I'm trying to actually work through. So for scaling from the single cell to the populations, I will actually consider basically a McKendrick partial differential equation type approach, and basically describes at the macroscopically what's happening, and then this is going to be actually scaling to the number of the individuals carrying these traits, right? So this is in general framework that you can apply to stochastic traits, how they are actually scaling to the population level. And this is the H operator is actually keeping the stochastic behavior of the traits and including the sort of like the mortality, how you kill the individuals. And then by actually imposing sort of like boundary condition, so you can also work through sort of like reproductive parts. But most of the time, these kind of partial differential equation approaches actually fail because they are actually becoming very difficult to solve. And practically what we do is just we then use sort of like a matrix kind of algebra. And this is quite often known as like sort of like age and stage structured actually population models, right? But there is actually these two framework actually are consistent with each other, right? And then all this sort of idea is then to construct a matrix transition transition matrix, and then try to understand your population dynamics from there. All right, so this is the general, the theoretical framework I will be using, but now I'll maybe go back to a little bit more the reality. So I will be interested in bacterial aging, and I already maybe hear that some of you are asking this question, whether do they actually age or not. So as actually this is until very recently, so we didn't know that actually bacteria were aging, but they do. So it's actually, this is starting from 2005, just by looking at the single cell level. They follow the sort of like the dividing the bacteria. And also they're following sort of like what is the old pole and what is the new pole. So whenever there's a division, of course, in the middle, there's a newly synthesized pole, and you can actually keep track of those old ones, right? So here labeled as like with the blue and red, right? At least in an agar plate, sort of like you can follow it for 8 generations. And then you observe typically this behavior, sort of like you realize that there isn't some imbalance there. So it actually shows that actually those who are carrying the old pole, they're actually getting slower in division. And then later on also there has actually been done, as I will show also that there are a bit longer experiments actually thanks to this sort of like what we call the mother machine. So in this kind of setup, you can actually trap the cells in one side is closed, and then what we call a mother cell. And then whenever there's actually division, there's actually daughter cells are washed away, but you keep the mother cells. And then you can run very long experiments, a few days, maybe even until a week. Yeah. And this allows you actually to collect individual data, thousands of them. And what is nice that they are the same environment and they're isocolonic, so it's like they're the same genetics. So it's like you try to understand sort of like the stochastic variation, mostly driven by actually the aging part. And then we can, as you see, we can segment them, track them, and then also get physiological data, also like gene expression data, and look at actually all this together. But there are actually two things that actually I would like to highlight here. So it's like for this damage dynamics. So there is actually for bacteria there are two things important. One is stochasticity, and this is what we also see when we look at, for example, the gene expression data. So this is at the first row showing you when we look at the RPUS gene expression profiles in any single cell lineage, you see that actually it's quite stochastically is actually changing, and this is typically it is so. So this is maybe suggesting that inside the cell, at each individual cell, maybe there is actually quite stochastic environment, and also for this is true for the damage. We also try to understand the damage characteristic when there are actually divisions. So when there are division and when we have mother and the daughter cell, what we observe is when we focus on the protein aggregation data. And this is also considered as like a cellular damage, that actually there is sort of like big asymmetry. So that the mother lineage conceives actually more protein aggregation, and they are then actually driven to be a little bit losing their fitness. Okay, I will be considering especially this stochasticity and the asymmetry, and try to understand how they're actually affecting the population dynamics. But first, I would like to do this in some modeling perspective. And some minimal model that we can come up is just writing down a jump diffusion equation, which is a stochastic differential equation. That is actually the type that I'm writing now is just basically a geometric Brownian motion. The first two terms having the deterministic rate epsilon here, sigma, telling you the strength of the noise, which is described as like Wiener process, right? And just adding the last part is actually allowing you to actually making big jumps, right? Okay. And this is maybe the minimum sort of like stochastic model you can maybe think of. And previously, sort of like we tried to study this in a cohort environment, starting with maybe 100, let's say, individuals. They all start at the same damage level, and where do they actually end up, right? So just didn't actually say one thing more about the model, sort of like I was trying to put. So there's also like a critical damage level. So when there is actually this trajectories, individual trajectories hits this damage, maximum damage, actually this defines the cell death, okay? And what we looked, so what we try to do is sort of like making some approximation based on division rates are Gaussian distributed. You can actually make an approximation to this jump diffusion equation and make it like more like geometric Brownian motion, so that you can actually come up with the lifespan distribution and survival curve, right? So and you can write down analytically, and it shows that actually the comparison to simulations from the exact jump diffusion equation shows that this is a good approximation. And maybe more importantly, it's using this kind of more age-driven, let's say, mortality model sort of fits quite well existing data sets. So this is showing you 4 different data sets, all are E. coli. So it's like, again, from mother machine, and typically showing here a wild type and in comparison to a red curve, which is an intervention, so most likely like a mutation event, right? So these mutants probably creating some more stress, and you see that actually the mortality rates are increasing. But other thing that's typically what you see is that they are like starting with a basal level of mortality, and then after some time, typically is like at the first day, they are starting to increase. So they are actually being aged at this time, and then they are reaching a plateau. Okay. This is one way of maybe showing that the bacteria is also aging at the mortality level. Okay, and this project, so we would like to actually now understand, not just looking at the cohort, but now how can we actually project this to the population dynamics? All right, so, and this is, we will put this into sort of like a age stage structured population model framework. And as we have now these analytical expressions for transition probabilities and also survivals, this can be done easily. And this is showing you basically two example cases when there is the damage parameters are, let's say, not very high, and then when damage parameters are a little bit higher, red versus blue, and it's not a perfect approximation, but it actually catches sort of like the patterns that we observe. So it's like you see that actually the population dynamics will be slower when there is a stress, when there is actually more damage rate building up. And very nicely, maybe that's also at the population level, we actually catch sort of like the death rate, okay? And using sort of like this metrics part, sort of like metrics model, so then we can ask actually this larger question. So it doesn't show it here, but it's actually the X scale is like a noise. So I increase the noise in my macroscopic level damage dynamics, and I change sort of like my asymmetry level, again, when there's a partitioning, and how much actually these two parameters will affect the population dynamics. And I show you three different examples. The first one shows you when there is actually not much damage rate, right? And this is typically then you expect, then there's not going to be much mortality due to damage or aging. So then the population will grow as you expect from the division rate calculations, right? This is, yeah. And when there is intermediate, then that's going to be actually now the stochasticity and the asymmetry will play important role, right? Even when you increase the damage so much high, so most of the parameter space is going to give you actually the zero or even negative population growth, right? But you might see this, maybe you can think of sort of evolutionarily, maybe there might be some strategies. If there is such strategies, you can see that maybe cells can switch to more asymmetric level in order to actually run away from extinction. Yeah? And this actually might give you some idea about maybe the evolutionary level, why actually these microscopic damage parameters might be important. All right, but I will not go so much into the evolutionary level much. The last part of and the remaining part, I will just go into our experimental data. So we try to actually look at this also at the experimental level. And what we did, and this is very loaded figure, but at the first row, we have the single data, sort of like I described mother machine. So, and then red here is referring to a stress actually condition, which we achieved by not providing glucose to the E. coli, right? So, so that means glucose limitation referred to rate data, stress condition, and the blue one is the standard condition, standard media provided to E. coli, right? So what you see that the division rates are reduced, but not, they don't grow really to zero, but a little bit reduced. What you are drastically seeing that actually the mortality rates when there's a stress is very quickly going up, right? In comparison to the blue case. And you see that RPOS, which is the stress response signal in E. coli, is higher than the standard case, okay? And at the same time, we can do with the same conditions, the experiments in plate reader, which is basically population data. Now you see that actually the stress, when there's a stress, the red curve is actually down. There is actually reduction in the population growth, which you would expect. But most of the time, this is being modeled considering sort of like the density regulations, right? Typically like logistic growths, etc., which is probably playing an important role. But most of the time, people didn't actually so much study sort of like how much actually age-driven mortality is now playing a role to reduce the population growth. Indeed, by adding prometium iodide, like we are doing in the single cell data, we can look at the cell lysation event telling us about mortality in plate reader. We also see that actually they are actually very much quickly arising and in comparison to the non-stressed cells. And similarly, like RPOS is also data is much more elevated. All right, so this is, you see sort of like there is an agreement between the single cell data and population data, but actually, can we actually use sort of our model to understand it? So what we can do, we can indeed, this mortality rate data showing you in the first row, showing you this the curve, which is actually fitting of our damage model, which I just described. And from here, we can extract the damage parameters. And we can also look at the RPS data for asymmetry. And we can use this information. There's only one thing is remaining as a free parameter, is the scaling parameter, which is the scaling for the time. And this is the only thing is a free parameter. We can maybe search for the best result and then look whether actually such damage model catches sort of like the population dynamics. And the results is showing you here. I'm only looking at very early time, so that actually, so it's not so much density regulation is starting. Typically, this is considered sort of like the half of the carrying capacity. And you see that actually this damage model capturing sort of like this reduction in population dynamics. And not only that, but also sort of like, at least qualitatively, it catches sort of like the death rate and the mean damage, yeah? And we can at least theoretically quickly test whether this is because of the damage parameters we are estimating. We can just swap, right? We can swap the damage parameters, keeping the same division rates, yeah? And then you see, for example, in this comparison, this is the swapping in the last row. You actually lose sort of like this scaling to population dynamics. Even you are actually reversing the effect in the death rate and mean damage rate, okay? So this is at least supporting that. So this is what we are doing with the model is at least partially is catching what's happening in the population dynamics. All right, so this is what I wanted to actually tell you a little bit. So what I wanted to tell, so especially the cellular aging damage kind of dynamics are often overlooked when we are scaling to population dynamics. And this is maybe especially important when there are stress in the environment. And this is probably very important when there are, for example, like drug applications, right, to bacteria. Yeah. So I try to show you the sort of like looking at from the modeling point of view and also have to do it with the experiment and it shows at least some partly good understanding of what is happening when we have like applying a stress to glucose limitation in our case, why we have like population growth reduction. And a little bit outlook, I mean, so it's like, I didn't so much talk about an evolution, but at least this framework could be used at least in applications like a drug-resistant evolution. Because most of the time that what we consider, so when you apply a drug, sort of like antimicrobial, so you see like very drastic population decrease. But what happens so that there are some persister cells still remaining, those you can think of like they are the ones who can prolong their lifetime, like they can run away from mortality. And then this maybe gives them enough time to catch the mutations. And then with these new mutations now coming, they can actually develop sort of like the resistant evolution. And in this framework, I'm actually talking about constructing another projection matrix, right? Now the details are a little bit different inside the parameters because they are maybe behaving different. They might have maybe changed the stochasticity level of the damage. This can be regulated, if you think, or asymmetry. And this changed the transition matrix. And the question is, can we actually catch this kind of sort of like data, at least in this framework? All right, so this is what I wanted to tell. I would be very happy if you have questions or any feedback.
One question.
I was wondering, like, you consider symmetry in the damage spread, but I was wondering if you could also consider symmetry in cell division size, for instance. You have two different sizes.
I guess, yeah, I didn't show it actually here, but I do see actually this size asymmetry as well. Here, what I showed to you is that there's RPOS, total RPOS is different when they actually divide. So mother one is getting more RPOS signal, probably due to maybe the damage. But it's also like the mother one is actually the bigger in size. And this is what I already showed you in our different work, where we are actually focused on the protein aggregation. This is also what we see there. So the mother is slightly bigger. So it suggests maybe that the protein aggregation is an important damage in bacteria.